So next, if you still remember what we have gone through just now, okay, on previous lessons, we talked about this evaporations, everything, transpiration occur in the pathway number 1, okay, step number 1, step number 2, from the xylem vessel elements into the mesophyll cells. So now we look at how the water molecule actually enter into the root hair cells from the soil. Okay, so generally, a root hair reach into the space between the soil particle to absorb the water molecule. The process again is by passive. So water molecule move in the root hair cell, again highlight down the water potential gradient. Generally, soil water will have high water potential because relatively dilute solution. Cytoplasm and cell set inside root hair cell has low water potential because of the dissolved inorganics and organic substances. So due to this, they have a steep water potential gradients, allowing the water to move into the cytoplasm of the root hair cells by osmosis, okay, down the water potential gradient. Okay, so if you look at these, the absorptions here take place. You can see that this is the root hair cells. So the root hair actually, the adaptation, guys, you can see that they have the root hair. The epidermal cell at the root, you have the root hair cells. So they have the very thin permeable cellulose cell wall. So why thin? Why we must have the thin permeable cellulose cell wall? So again, they have the short diffusion pathway. Okay. Diffusion pathway. So depend in water, so short osmosis pathway. Okay, water that I prefer to put it as the osmosis since we talk about the water. So very short osmosis pathway. Okay, so allows the water molecule to enter into the cytoplasm of the root hair cells. Okay, so we can see that we have the adaptation here. Large number of fine root hair cells provide the large surface area in contact with the soil water, so increase the rate of water absorption. So root hair cell also important for the absorption of the mineral ions. So it depends the concentration gradient. It can be facilitated diffusion. It can be active transport. And we do know that even though active transport involved here, okay, it's because accumulations of the ions. When the ions concentrations now increases, then it's going to lower the water potential in the root hair cells. Therefore, osmosis follow. But because of active transport require ATP, the involvement of ATP, but generally the transportation of water molecule from the soil into the root hair cells, we still claim it as passive process because we don't purposely, the plant won't purposely to take in the ions so that it can take in the water molecule. They do not need to do this. Are you clear? They do not need, because by natural process, the root hair cell will have more or higher water potential compared, the soil will have the higher water potential compared to the root hair cells. Okay, so we are done for this part already. Then from the root hair cell to the xylem. So from the root hair cell to the xylem, so the regions is termed as the cortex of the roots, and then enter into the xylem. So it means that they have to cross these cortical cells. Okay, so the process is by passive process, means, okay, passive basically means do not require, okay, the process does not require ATP. Okay, so it means that the water potential in the xylem vessel now is lower than the water potential in the root hair cells. So therefore, water molecule move. Okay, later I will do the summary for you guys. So the process again by down the water potential gradient. So the water molecule can take these two routes through the cortical cells, again, apoplastic and symplastic. As I said that, again, apoplastic pathway, cell wall to cell wall by diffusion. Symplastic pathway, the water molecule will move into the cytoplasm, vacuole, or plasmodesmata. The same thing happen again along these cortical cells. Okay, so I will tell you guys later. So once the water molecule is special, endodermis, highlight endodermis, because endodermis will have this waxy band which is impermeable to water molecule called submarine. Are you clear? So you can imagine these submarines, okay, form these Caspian strips. This submarine actually form the band. So this band is called Caspian strip. So which form an impenetrable barrier to the water molecule. So means the apoplastic pathway now is blocked. Only symplastic pathway allowed. Are you clear? Only symplastic pathway can allow. So means that you can see that now. This is the endodermis. So due to the depositions of the submarine, so this submarine, can you see that it form a band? It's not like it happened here in entire. So it's something like you try to imagine. So you try to imagine that you have a box. Okay, you have the box, okay, and then you tie a rubber band onto the box. So it means three-dimensional. Can you see that? You put something like block it. So when you put something like block it, means that apoplastic pathway. Now, again, remember, submarine actually deposit within the cell wall. So means that any pathway that requires cell wall won't be able. Can you see that? Won't be able to pass through. So this Caspian strips are very, very important to change the direction. Basically, apoplastic pathway have no choice now to enter through the symplastic. So means that they have to go through these passage cells, enter. Can you see that? So it block this Caspian strip, block apoplastic pathway. So the only way for water molecule to pass through, they have to go into the cytoplasm by symplastic pathway, and this pathway require the water molecule together with the ions to go through or to cross cell surface membrane. So now, in general idea, this is a checkpoint. Why I say checkpoint? Because if we don't actually do a blocking like this, any kind of ions actually can go through. If we're going to bring all the ions, are you clear whether the good ions, bad ions, whatever ions that we need or do not need, also will enter into the xylem and transport to the shoot. Are you clear? So in order to do a selections here, cell membranes is a selectively permeable. So it means that for this apoplastic pathway, we have to stop them. The plant have to stop the apoplastic pathway to make sure that not all ions will be carried by water molecule, or carried by water, enter into the xylem and transport to the shoot. Because sometimes root also needs the ion. We cannot actually lose too much of the ions to the leaf there. Are you clear? So it means they have to do the selection. So how we do the selection? Apoplastic pathway, we cannot do a selection at all because no cell surface membrane, cell wall actually fully permeable. So in order to do the selections, we have to mix now the movements or enter into the cytoplasm. So within the cell surface membrane, we do have the carrier protein, we do have the transfer protein to check. Okay, if let's say I don't want the calcium ions to move into the xylem, then very simple, we don't want calcium ion, we just no need to actually, the plant just no need to have the calcium ion channel. Okay. Now, but the thing here is this endodermal cell, when get older and older, the submarine now deposition become more extensive. So just now I say the apoplastic pathway blocked, right, by this brown submarine. So only the the symplastic pathway open. But sometimes these Caspian strip, the submarines, okay, continue to develop and then fill up all. Can you see that? Fill up all the cell wall. So it means that this cell no longer can allow the water molecule to pass through. So when plant go older and older, the deposition become more and more, it will block this. Okay, the whole cells, even symplastic pathway also cannot pass through. So symplastic only can pass through this what we call the passage cell. So passage cell are some of the cells keep the symplastic route open. Are you clear? Because sometimes when enzyme is there, the process will continue. Means the plant cannot stop it. So it's like something like chain reactions. Once you start it, you cannot stop. So at the end, you can see that they will stop the symplastic pathway as well. So you can see that, okay, figure 8.19 show you guys that the water taken up by the root hair cell crosses the cortex of the root and enter the xylem in the center of the root. So you can see that's how the apoplastic continue. Now, halfway through, they can change directions, guys. It's not like, okay, the water molecule know, oh, I died, must be apoplastic pathway. So you can see that apoplastic pathway will continue, but when you reach the Caspian strip, as I say that, you're going to block the apoplastic pathway, then you can see that now this the water molecule have to change the mode of the transportation. So from the apoplastic pathway change to symplastic pathway. So symplastic pathway will continue, no issues. Can I see that? Continue all the way into the xylem vessel element. Okay, so now I'm going to draw more detail pathway so that you can understand the entire process. Okay. So I'm going to summarize the part 3 and part 4, where we can see the water molecule now move from the soil to the root hair cells, and then from root hair cell to the cortex, and then cortex to endodermis, pericycle, xylem vessel elements, okay, at the roots. So before we start, let us look at the cross sections of the root. Okay, so transverse sections or cross section of the root. Okay, so let us look at this. So first of all, you can see that the roots, you have the root hair. Yeah, let me draw this way easier. Draw large enough. So you can see that we have some parts, okay, root hair cells. So I don't draw all, okay? So you can see that it's a root hair. Doesn't mean that they only have 4, okay, guys? Okay, so the first layer is called epidermis, which form this root hair cell. Yeah, it's very textbook drawing. Okay, so what is the first layer of the cells? So first layer of the cells here is termed as the epidermis. Okay, so consists of what kind of cells? Root hair cell. Okay, then between the epidermis and also the center ring here known as endodermis. Are you clear? So this layer is termed as endodermis. Oh, sorry. So point to the center. Yep. Okay, endodermis. Okay. So between the epidermis and also the endodermis, we have these regions known as the cortex. Okay. So it's also called cortical cells. Cortex or cortical cells. So it's formed by parenchyma. So ground tissues called parenchyma. We have learned previously. Okay, the parenchyma. So if we zoom into the endodermis, where we can get our vascular bundle. So you can see that the vascular bundle shows something like the star shape. Okay, and also between the arm of these star shapes, you have the phloem. A very textbook drawing. In fact, in the paper 3, we're going to learn. Okay, so you can see that the regions here, not the vascular bundle. So we do have the pericycle. Okay, we do have the pericycle, and then this one is the phloem, and this one is the xylem. So both of them form the vascular bundle. Okay, so so these are transverse sections of the root. So we start from the part 3. So we have this root hair cell. Okay, we had the root hair cells. So with the cell wall, always remember plant cells must have the cell wall. Okay, so outside here will be a soil particle, or say soil water. Easier. Okay, soil water. And this is the root hair cell. So it depends on directions of the water. Okay, movement, move. So generally, this part is a cytoplasm. So we do know that the water potential is lower in the cytoplasm. Soil water potential is higher. Okay, more dilute. So therefore, water molecule now enter. So water molecule can enter directly into the cell wall and move along the cell wall. So this one we just ignore. Are you clear? We just ignore. But we're more concerned about the water molecule that enter into the cytoplasm of the root hair cells. So this is the first process. So what's the first process here? So first process, okay, is the osmosis. So how we describe? So we say that water molecule move from where to where? From the soil water, from the soil water okay, to the cytoplasm of the root hair cells by osmosis down the water potential gradient. Are you clear? Okay, so in this case, I want to highlight a few things here. So it's by osmosis. But here's very important part here. Okay, highlight cytoplasm, because water molecule can enter the cell wall, but enter the cell wall is not osmosis. Enter the cell wall is by diffusion. So that's why I purposely put in the word cytoplasm. So by osmosis, because we concern more to this route. Okay, so water molecule enter. Okay, so then we do have the cortical cells. Okay, so we have cortical cells. I'm going to draw the cortical cells. Sorry, guys, let me draw. So these two cells, they are the cortical cells. Okay, I mean it's more than that, but just I use two to show. Okay, now so water molecule now can enter which way? So water molecule enter, it can continue move within the cell wall. So we move within the cell wall. So this is process number 2. Okay, cortical cells also will have the vacuole. Okay, they do have the plasmodesmata. So same thing, water molecule have 3 choices. Either within the cell wall, continue to move. Can you see that? Within the cell wall, continue to move. Okay, or move into the cytoplasm. Can you see that? Or move across the vacuole. So this is number 3 and number 4. So sorry, I don't want to go through a detail again for number 2, number 3, and number 4. I just summarize here. Number 2 is apoplast pathway. Okay, number 3 is the symplast pathway. And number 4 is the vacuolar pathway. Are you clear? Okay, so Sym plus and molecular pathway, we can group them together as Sym plus. Okay, so now we come to this, what we call the endodermis now. Okay, this is the endodermis. So by endodermis, so because of the depositions of this submarine from the Caspian strip, so block O, can you see that? So the presence of this submarine forming these Caspian strips, so means that water molecule now cannot, impossible for the apoplastic pathway. So the only way is by symplastics now allowed. Are you clear? So this part, I'm going to put it as number 4, number 5. So number 5, you can see that because due to the presence of submarine that form the Caspian strips, so apoplastic pathway is blocked. Allow only symplastic pathway. Are you clear? Okay, so endodermis. So after endodermis, you do have the pericycle cells. So this one is a pericycle. So it means that when water from the endodermis so enter, so it can continue with the apoplast or symplast pathway, doesn't matter now. Okay, because we have to control what kind of the ions and cells water, I mean what kind of ions can pass through already. So next, finally they reach into the finally now you can see the water molecule enter into the vessel element, xylem vessel elements. So with thin wall, thin primary wall, and also the lignification. Okay, so water molecule have to go how to go into the xylem vessel element in this case. Okay, so number 6 here. So number 6, okay, sorry, just now I said, okay, number 6, it can be apoplastic, okay, or symplastic, doesn't matter now. Okay, but for this water molecule to enter into this xylem vessel element, then they have to pass through the pits. So number 7, very important. So water molecule enter into the xylem vessel element. Okay, through the pits. Okay, so overall process down the water potential gradient. Okay, so once inside here, what actually happened? Can you see that? Because in this part, okay, I don't use an arrow. Okay, okay, this part. So due to the accumulations of water, so means that increase in the volume. So when increase in the volume, then increase in terms of the hydrostatic pressure because accumulations of the water molecule. Okay. So if you look at this water potential, if you compare high to low, Okay, high to low. So the first one, the highest actually is soil water. Number 2, followed by the root hair cell. Okay, number 3, dense cortical cells. Number 4, endodermis. Number 5, okay, pericycle. And number 6, xylem vessel element at the roots. So in this case, you can see that soil water will have the highest water potential, and xylem vessel elements have the lowest water potential among these 6 structures. So again, you can see that entire process down the water potential gradient. Okay, done. Okay, so what actually happened along the xylem? Okay, so along the xylem, we have to understand that this one mainly because of the hydrostatic pressure. Okay, so what actually happened? The removal of the water molecule from the xylem vessel at the leaf there reduces the water as a hydrostatic pressure. So what is hydrostatic pressure? Hydrostatic pressure is the pressure exerted by a liquid. Okay, because we remove, so therefore hydrostatic pressure at the top of the xylem vessel elements, this one near to the leaf, okay, will be lower than the pressure at the bottom, means that at the roots. Okay, at the shoots and the roots. So the removal of the water molecule into the xylem from the root hair cell now increases the hydrostatic pressure. So this pressure difference causes the water molecule to move up the xylem in a continuous column. And this column actually, okay, sorry, the movement of this water column is by the process called mass flow. So that this means that the water molecule together with any dissolved solute move together in a continuous column. Why can be continuous? Because there is no end wall. Clear not? There is no end wall of the between the columns, yeah, between the vessel elements, right? Okay. So which theory actually we're going to use here is the cohesion tension theory. It's a theory that we use how. Okay, it's the main way in which continuous column water move up the xylem vessel element. So first of all, what is mean tension? Tensions because, okay, the xylem vessel elements under the tensions because of the wall may collapse inwards because of the pressure difference. Okay, I will explain it later. However, due to this lignified cellulose cell wall, so this lignified cellulose cell wall can prevent the collapse of the xylem vessel elements. Okay, strengthens the wall. So what is mean cohesion and adhesions that take place? So if you look at the process here, you can see the water molecule move as a continuous column. So water molecule actually form the hydrogen bonds with water molecule by this cohesion. Okay, the cohesions. And what is the adhesions? Adhesions basically is the hydrogen bonds that form between the water molecule and also the cell wall. Okay, so this part, you need to be extra careful when you write it. Okay, now all the water molecule move together as the water molecule attracted each other by the cohesion force, and also water molecule are attracted highlight lignified cellulose cell wall. Now, you cannot use the word lignite, not lignite, but it's a lignified cellulose cell wall, or to be more easier, I mean, don't need to be wrong, you actually remove the word lignified. You just say that cellulose cell wall, then correct. Are you clear? So because it formed this bond, so this bond is a hydrogen bond formed between the water molecule and the cellular cell wall. So the force is termed as adhesion force. And we do know that the diameter of the xylem, okay, still remember, between 0.01 millimeter to 0.2 millimeter. So this is very, very small, okay? In the range is considered small to prevent the airlock. In case airlock take place, it will result the breaking of the water column. You cannot maintain the water column already. You try to imagine that between, I run whole hand in between, I break it. Okay, so by forming this bubble. In case airlock form, don't worry, we still have the pits that allows the water molecule to move out from one vessel to another to bypass the airlock. We have learned this previously. Okay. So let us look at the part 5 now. Part 5 along xylem vessel element. Okay, part 5 along xylem vessel element. So what actually happened here is, so 3 things that we need to learn here. First of all, this is a xylem vessel element. Okay, in general. So this is the part that we say that's near to the shoot. This is a xylem vessel element at the root. So we do know that these vessel elements, they form a continuous column. Okay, due to the breakdowns of the, sorry, let me draw it again. Yeah, I just draw it a quick one. And we do know that they do, okay, they do have this lignified part, and okay, some parts that without the lignin. Okay, so unlignified cell wall forming the pits. Okay, so what actually happened here is, now at the shoot, what actually happened? Due to the transpiration. Correct or not? So it means that removal of the water. Okay, so means that at the shoot, the hydrostatic pressure is low because you remove, okay, the water molecule out. So at the root, what actually happened? Due to the entry of the water, correct or not? Because from the soil, water molecule move from the soil to the xylem vessel element. And due to this, because of accumulations of water molecule, okay, so sorry, at the shoots, what actually happened? At the root. Okay, so therefore hydrostatic pressure is high because due to the accumulations of the water molecule. So therefore, can you see that in this case, there is a pressure gradient. So this pressure gradient is termed as hydrostatic pressure gradient. So what actually happened here? So high pressure, liquids will always move from the high pressure to low pressure. Due to this, we call it a transpiration pool. Are you clear? So it creates a tension. So to create a tension, so this low hydrostatic pressure, so the collapse of the column is prevented by the lignification. Can you see that? Okay, the collapse of the column is prevented by the lignification. Okay, so how the water molecule move in this case from the high to low, okay, down the hydrostatic pressure. So this process is termed as mass flow. You can see that it's not one by one, but it's a whole column of the water molecule. Are you clear? Forming a continuous column anti the gravity. Okay, so we do use this term called cohesions and tensions. Cohesions tensions theory. So what it means by the cohesions? So in this case, the force that maintains the column is the cohesions and adhesions. So what's the cohesion? Cohesion is the bond formed, the hydrogen bond. Okay, form. So for cohesions between water molecule. So maintain the column. But maintain column not enough. We want to be, continue to move up. So they need to have the adhesions. Okay, so this adhesion is a hydrogen bond formed between water molecule and the cellulose cell wall. Are you clear? So these cohesions and adhesions. Number two, we say that the column also under tensions. So why under tensions? The column is under tensions. So due to transpiration. Cool. So due to these two factors, now water molecule actually move by mass flow from the xylem at near to the roots all the way to the xylem at the shoots. Okay, so due to these cohesions and tensions that created, okay, and the tensions created during the transpiration pool, and also the cohesion adhesions because of the formations of hydrogen bonds that between the H2O or water molecule by cohesions between the water molecule and the cellulose cell wall, which is adhesions. Okay, so that's all for today.